Hello everyone, my name is Muhammad Asib and I'm working as a senior researcher in S&K Therapeutics. Before going into details, first I would like to explain give a short overview of the S&K Therapeutics. S&K was established 2 years before by Professor Sang-Dan Choi. He is a renowned professor in the field of immunology. Professor Sang-Dan Choi is a renowned professor in the field of immunology. He published 250 research articles in the field of immunology and also filed a number of patents in the field of immunology. Our goal is to develop efficient therapeutic targets and therapies for the treatment of autoimmune diseases. This is the content of my today talk. So first I will give a short overview of the autoimmune diseases, and then mainly I will focus on the core technology that S&K developed so far, and then I will explain about the core competence that our competitors, and then the pipeline that we are going to further develop in the future, or our future goals. Here is introduction. So what is autoimmune diseases? Like the SLE is and the psoriasis is one of the main important autoimmune diseases, and which cause several number of people are affected because of these autoimmune diseases. Actually, the autoimmune diseases, it's because of the hyperinflammations, and due to autoimmunity, it exhibits this rashes on the skins and the oral ulcers and several different symptoms like arthritis, and it's appear at the skin here. There are more than 5 million of people worldwide they are affected because of the lupus, and most of them are the women. Why the women are so important? Like they suffer the women are suffer because of it, because in women they excrete hormonal changes. Because of it, they are more suffering, and it's affect the woman childbearing age at the age of 5 to 10% mortality rate. In Korea, it affect total 30,000 of people were diagnosed because of the SLE. If you look at the market of the lupus, so in 2015, the data were reported by the global lupus market. It was around $1 million. Now it is expected to be increased up to more than $2 million by 2025. So here, if we can see that the analysis like the global lupus market, so these are the immunosuppressant that are reported, which is very higher reported up till 2027 for the lupus, as compared to other anti-malarial drugs, corticosteroids, and other biologics like antibodies, etc. So the growth rate is very higher, and it increased up to 10.8%. And there are very important needs to develop the lupus drug that is cost effective for the severe conditions, and as well as that to decrease the incidence and mortality rates, to improve the patient life. And here, these are the number of main players which are involved and to develop the drugs for the lupus. The current status of the lupus treatment, that is like there are a number of drugs which is reported, that is like hydroxychloroquine, the Benelista, which is one of the antibodies in the market, but all of these drugs are very expensive. So normal, like the patient that cannot accommodate and to purchase these kind of drugs, which is not easy to access for every kind of patients. So there is an immense need to develop the small molecule drug which is cost effective and to have greater efficiencies. So here they only relieves the current drug, they only relieves a number of like the basic symptoms, not to totally recover the SLE. So, and the other problem that in antibody, like in such in Benelista, that is a low compliance with injections. So there is immense need to develop the low molecular weight compounds that have higher potency, which is inexpensive and taken as oral drugs. So the data that is reported by the global lupus market in 2020, So the market for the oral drug, that is very higher. So in the blue line here in the graph shows the increasing of the oral drug demands in the market up to 2027. And the main problem, that is the socioeconomic losses because of the current available therapy for the lupus, that is the women childbearing age negatively impact economic activity, and it's very alarming for those countries which have the less number of populations and their growth. And the other main problem, that is very expensive. So we need to develop a drug which is less expensive as compared to the reported drug. I will briefly explain about the S&K core technology that we developed so far and our targets. So we developed our strategy that we used for the development of the SLE drugs. We targeted here in this diagram, if you can see that the TLR which are present at the PDC, upon activations, it activate the dendritic cells and the B cells and the T cells. So here it activates different cytokines, like for example the interferons, the interleukin-17s, and many others, which are mainly involved in the systematic lupus, psoriasis, and other autoimmune diseases. So our aim is to target, and we targeted this toll-like receptor 7 and 9, which is present at the endosomes. And these are the number of drugs which are reported for the TLR, which are working as a TLR inhibitors. Most of them are the oligonucleotides. So if we block here the TLR signaling, so downstream it can inhibit the several, like the IROC, which is one of the important IROC kinases, which is involved in the lupus, in the cancers, and many other diseases. So this particular target is very important, and upon targeting it, it have a great potential to overcome the several autoimmune diseases. So further we checked, and to prove our concept, that we took like the lupus mice model. There are two different kind of mice model we use in our study. One is the CAE57, and the other one is MRL mice model, lupus mice model, that we used for our studies, and we treated by the S&K product. So here if you can see that it overcomes symptoms and inflammations which is caused because of the SLE. So to further strengthen our drug move toward the drug candidate and toward the development, we further checked the different cytokines which are critically involved in the systematic lupus and autoimmune diseases. So here are the two different kind of mice, lupus mice model which we used, and this is our compound TEC5A that we used. So it significantly reduced the inhibition, or significantly inhibit the interleukin-17 cytokines by our S&K product, that is TEC5A, and it reduced the lymph node. And here if you can see that it also inhibit the MyD88, which is the downstream, and the TLR9, the TLR7, and different other cytokines. Further we move toward the lead optimization process. So we optimized our lead, that is the TEC13 to TEC15s and the ETI 24 to 52. So initially the activity of the TEC, our initial lead candidate, that is 2.7, and so we improve for like ETI 24, and we improve its LC50 and like the toxicity. We improve its inhibition for ETI 41, which is 0.63, which is quite good as compared to the initial lead. Here you can see that there are the MRL lupus mice model. Here you can clearly see the rashes on the mice. And then upon treatment of our S&K candidate ETI 24 were treated. Upon treatment, it significantly reduces the rashes on the mice. To further optimize our lead candidate, we took hydroxychloroquine here as a control, which is in the market and FDA approved for anti-malarial, but it ought to work for the SLE, but not in the severe case. It's work only for the initial SLE. So here the LD50 for the hydroxychloroquine, that is 52, while the IC50 is 3.20. As compared to this, our compound, the ETI41, its LD50 is quite good as compared to hydroxychloroquine, and the activity and the efficiency is also good, is 0.63 IC50 value, and other compound as well. This is for the TLR7, and in the TLR9 it's also like here the ETI41, it saw a very good efficacy, and it shows that 0.16 IC50, and the LD50 is also good as compared to the hydroxychloroquine. Here you can see that there are the number of our lead candidates and its activity in the toll-like receptor 3, 7, and 9, which reside in the endosomes. To further check the activity of our compound, we took hydroxychloroquine 60 mg per kg and our compound ETI60 to ETI41, these three candidates for comparison. So we checked here in HCQ upon treatment of 60 mg per kg. So it's still there are the SLE symptoms present in the mouse which is treated through HCQ. Why HCQ? While as compared to this, the ETI60 and other our compound ETI66, the SLE infection or the SLE symptoms were significantly reduced in all of our three candidates. Further, we took the HCQ while taking the 15 mg per kg. In previous slide, we took HCQ 60 mg per kg, while our candidates, the ETI series, we took 30 mg per kg, which is the half of that. While in that condition, in this slide, we compare our candidates by saying like administrating the of these three compound while in similar amount, like that is the 15 mg per kg. So here you can see that the HCQ, the symptoms of the SLE were not completely recovered, or even not recovered because by treating the HCQ, as compared to this, the ETI60, which shows, and ETI41, which shows a greater efficiencies as compared to HCQ by even at 15 mg per kg. So if we can consider the dosage, so HCQ dosage is 200 to 400 milligram, while as compared to this, the ETI41 is the best, and we can use even less than 50 milligram per 50 milligram. This is another mice model, and we further look and confirm in NZB mice to check the activity of our ETI41. So here you can see that this is the HCQ treatment, and here is the ETI41. So here the ETI41 significantly reduced the SLE symptoms and the infections, and it also reduced the swelling of the limb. So the pharmacological effect is one of the significant steps in there toward the drug discovery and drug designing. So here we tested our candidates for ETI41 and ETI66 to ETI66, our top lead candidates, to check its pharmacokinetics, to either it shows any toxicity or not. So here you can see that the ADMET properties are showing very promising, and it did not show any positive result, like it did not show any toxicity for all these three candidates. Further, like we check and we tested in two different kind of administration, like we took as IV and we took as an oral drug. So here it shows the area under the curve, which is one of the important factors of the pharmacokinetics. So it shows the number is pretty much good, and in both in the IV and the oral. And the half-life of our drug candidates is also good, which is around 4.7. It's pretty much good at this stage. So to further strengthen our to move toward the drug discovery, and we further tested the 468 different kind of human kinases to either to check if there is any side effect of our compound or not. So the result was pretty much good, and it did not show any interruption in the metabolisms and signal transduction. So here is the summary. In total we took around 82 new compounds, and we further checked the 3 optimal candidates, ETI 41, 60 and 66 for the future plan, and we are proceeding it as a future, proceeding it toward the drug development. And we also tested the mode of actions and for 30 compounds, and we are planning to for the drug formulations and efficacy. In this study we used like the 2 different kind of mice model, lupus mice model. One is the NZB and the other one is FAS LPR mice model, and the results was pretty much good. Now next is our plan to expand our compound testing toward different diseases, like toward the psoriasis, toward the rheumatoid arthritis, Alzheimer, and even in the COVID to check its antiviral activity. So here is some of the competitor that the advantages that S&K lead compounds that we have as compared to the reported Benelista antibodies. Our compound or our lead that achieve higher efficacy than Benelista, and reduce the patient burdens. And the other thing is that the Benelista, the compound that is the antibody which is larger in size, and that's are recommended, and like the people they are taking as injections, while and the target that is the BAF receptor, which is like at the downstream of the toll-like receptors. And the important thing is that is very expensive, so every patient cannot treat by or use that particular antibodies. So our objective is that, and we developed the small molecule compound which is small in size. Every patient can take as a pill in the home, and it's the main thing. We target the toll-like receptor 7 and 9, which is present at the upstream of the BAF receptor, and which shows the excellent efficacy. And the low cost, like its cost only approximately $300 to $500 per year, as compared to the reported or the currently available antibodies, which is much higher. So there are some of the competitive candidates in the clinical trial. So currently there are IMO 8400 and this one. These are the oligonucleotides, and which is in the clinical phase 2 for the SLE. So the disadvantage, or we can say that the disadvantage of this, it's oligonucleotide based. It's not a kind of like small molecule. Only 2 small molecules are reported up till now, and these are at the preclinical stages. So if we can compare our S&K candidates and our leads, so our compounds are not only efficient than the competing ones, but also inhibit the toll-like receptor 3, 7, 8, and 9, while having higher effectiveness and with full inhibition, and have lower cost as compared to the others. We not only here focus on the SLE, we further checked our candidates in the psoriasis, which is one of the skin diseases, and toll-like receptor 3, 7, and 9, they are present at the top, and it's activate the downstream like the different interferon, and the main key players, that is the interleukin 17 or 23 and 12, they are the main player in the psoriasis and other skin diseases. And IL-17 and TNF are also one of the important which is present in the psoriasis mice model. So further we checked to confirm the activity of our compound or our lead candidates in the lupus, other than lupus, that is the psoriasis. So here upon treatment of our compounds, like here is the psoriasis mice model, and upon treatment it overcome up to 80 to 90% relief to the psoriasis patients. To further check the different cytokines which are involved in the psoriasis, we checked the activity of ETI 41 and ETI 60. So here is the ETI 41, it's completely recovers the psoriasis, and you can see like in the control here is the redness on the skin, while skin surface, where both of our candidate treatment, upon treatment it's recovered. So here you can see that interleukin 17 was significantly inhibited by our both compounds. And similarly for interleukin 23. If we look at the psoriasis market analysis, so it was 9.3% and around 11.4 billions in 2016, but it is expected from the data which is generated by the lupus global market. So it is expected that it will reach approximately 21.4 billion in 2022. So although the market share of TNF inhibitor as well in 2016, but it is thought to be affected by increasing preference for interleukin inhibitors. And it's like the unmet needs and to suggest the drug and which are safe and are effective as oral therapy, which with the prices are less as compared to others. And there are a number of main players which are involved in the in the treatment of psoriasis, that are AstraZeneca and Johnson & Johnson, and the others main players are there. These are the number of current psoriasis treatment. If you can see here, so most of these candidates are the treatment or formulation is subcutaneous and IV, but only few of them are oral. So our goal is that to develop the small molecule which is cost effective, which have efficient results against these autoimmune diseases, such as particularly for SLE and psoriasis. And here the most of them, they are the antibodies, which are already mentioned that they are very expensive. So the next generation or the future drug development and the pipeline that is for the psoriasis are the interleukin 23 and the interleukin 17. They are playing a major role in the skin diseases, particularly in the SLE and the psoriasis. So it's very important to target these interleukin. And up till now, the reported compounds which are at the different stages of the clinical, these are, all of them are, like most of them, they are the antibodies, not the small molecule. Only few of them are the small molecule, but these are only one of the compound. This is on the small molecule, but it's not very specific towards targeting the toll-like receptor 2 and 4, but our target is the endosomal TLR, that is 3, 7, 8, and 9. So this is the indication that at the first step we checked the compound of our compound and the S&K lead candidates. So we tested in the systematic lupus and the psoriasis. Now further we are moving toward check it into different diseases like the rheumatoid arthritis, the steroid and asthma, and that will be the expansion for our second and third. We are challenging and we are trying to target the antiviral activity of these compounds, such as and to test it for the COVID-19 and the other anti-malarial and the respiratory diseases. So here I would like to thanks to our research team which are behind these successful results. So our collectively hope are that to develop the drug which is efficient, cost effective for the patient lives. And at the end, I would like to thank you to the organizer of today this events that to share our ideas and our development and our technology to share with all the others researchers. Thank you.